The genetic code. Human body cells contain 23 chromosome pairs or 46 chromosomes. Chromosomes are made up of many genes joined together like beads on a string. Although you have only 23 pairs of chromosomes, your body cells each contain about 35,000 genes, and each gene controls a trait. The main function of genes is to control the production of proteins in an organism's cells. Proteins help to determine the size, shape, color, and many other traits of an organism. Chromosomes are made of DNA or deoxyribonucleic acid. A gene is a section of a DNA molecule that contains the instructions of how to code for a specific protein. A gene is made up of a series of nitrogen bases in a row, and DNA molecules are made of 4 nitrogen bases: adenine, thymine, guanine, and cytosine. These bases form the rungs of the DNA ladder. A single gene on a chromosome may contain anywhere from several hundreds to a million or more of these bases. Each gene is located on a specific place on a chromosome. When a gene contains the code that determines the structure of the protein, it is in the order of the nitrogen bases along a gene that forms a genetic code that specifies what type of protein will be produced. Remember back from the cell chapter that proteins are long-chain molecules made up of individual amino acids? Well, in the genetic code, a group of 3 DNA bases code for a specific amino acid. The order of the 3 base code determines the order in which amino acids are put together to form a protein. The production of proteins is called protein synthesis. And the cell uses information from a gene and a chromosome to produce a specific protein. Protein synthesis takes place on the ribosomes in the cytoplasm in a cell. Now, as you know, the cytoplasm is outside the nucleus. The chromosomes, however, are found inside the nucleus. So how then does the information needed to produce proteins get out of the nucleus and into the cytoplasm? Well, before protein synthesis can take place, a messenger must first carry the genetic code from the DNA inside the nucleus into the cytoplasm. This genetic messenger is called ribonucleic acid or RNA. Although RNA is similar to DNA, the two molecules differ in some important ways. Unlike DNA, which has 2 strands, RNA has only 1 strand. RNA also contains a different sugar molecule from the sugar found in DNA. Another difference between DNA and RNA is in their nitrogen bases. Like DNA, RNA contains adenine, guanine, and cytosine. However, instead of thymine, RNA contains uracil. Now, there are several types of RNA involved in protein synthesis. Messenger RNA copies that coded message from the DNA in the nucleus and carries the message to the ribosome in cytoplasm. Another type of RNA, called transfer RNA, is going to carry that amino acid to the ribosome and adds them to growing proteins. The process of protein synthesis is shown here in this figure. Now, in the nucleus, a DNA molecule serves as a pattern for making messenger RNA. A DNA molecule will unzip between the base pairs, and an RNA base match up along one of the DNA strands. This genetic information in the DNA is transferred to the messenger RNA. You need to understand that the DNA never leaves the nucleus. The messenger RNA is now in the cytoplasm, and the pattern has been copied from the DNA. Now, when the messenger RNA enters the cytoplasm, it attaches to a ribosome, which is this large greenish blue blob here. This is where the production of protein chains begin. The ribosome moves along the messenger RNA strand. While moving, transfer RNA molecules carry specific amino acids to the ribosome. There they read the message in messenger RNA by matching up with 3-letter codes of bases. The protein chain grows as each amino acid is attached. This protein chain will continue to grow until the ribosome reaches a 3-letter code that signals it to stop. The ribosome then releases the completed protein. Sometimes during protein production, a mistake occurs in one gene of the chromosome. Now, instead of a nitrogen base adenine, for example, the DNA molecule might have the base guanine. Such a mistake is one type of mutation that occurs in a cell's hereditary material. A mutation is any change in a gene or chromosome. Mutations can cause a cell to produce an incorrect protein during protein synthesis, and as a result, the organism's trait or phenotype may be different from what it normally would have been. The example shown here throws three types of mutations that can occur in genes. The first is deletion, where one base pair may be removed. So for example, if DNA instructed the protein to say the red hat, with the deletion of a base pair, the message now reads the rec at. This may or may not cause an issue depending on which gene the mistake was in. A second mutation could be substitution. A single base may be substituted for another. So instead of saying the red hat, the message has been coded to say the red cat. And a third type of mutation that can occur is called addition, in which a base pair is added. As you can see in the example again, our message should say the red hat and instead now reads the reed hat. Because mutations can introduce changes in an organism, they can be a source of genetic variety. Some mutations are harmful to an organism. A few mutations, however, are helpful, and others are neither helpful nor harmful. A mutation is considered harmful to an organism if it reduces the organism's chance for survival and reproduction. All right, let's take a break and show what you know.